Good afternoon, and thank you everyone for attending on this late stage at the last day. And I'd like to thank the meeting organizers for the opportunity to present our technology. Regenerex is a late-stage allogeneic cell therapy company that is a therapeutic platform for the potential to treat a number of different disorders. It provides stem cell transplant to mismatched and unrelated donor and recipient pairs, thereby expanding the donor pool. The conditioning is non-myeloablative and performed as an outpatient, and the patients are managed as outpatient. We have an active IND approved for Phase III in Living Donor Kidney Transplant, which is our most mature indication. We have a GMP manufacturing facility in place that has been deemed appropriate for Phase III in commercial manufacturing, and we have very compelling results, which I'll show you, that have emerged from a relatively large Phase II study in living donor kidney transplantation, including up to unmatched and unrelated subjects. And that really opens the door for potentially an off-the-shelf therapy for non-organ transplant recipients, including autoimmune disorders. We have orphan drug designation and also RMAT designation by the US FDA. Essentially, the FCRx product induces donor-specific and recipient-specific transplantation tolerance, and that has major implications for both solid organ transplant recipients as well as patients with autoimmune disorders. And in autoimmune disorders, it resets the autoimmune system by replacing an unhealthy one partially by a healthy donor stem cell system. It involves bone marrow chimerism, and chimerism refers to the coexistence of two genetically different components in one organism. And obviously, the mythological figures are the best recognized, and our patients are chimeras in that they are hematopoietic chimeras. The technology is based on a cell that is not a stem cell. It is bone marrow derived, and it's a very tolerogenic cell. It's CD8 positive and T cell receptor negative and induces a number of regulatory cells in vivo. We have a very broad patent portfolio for the human facilitating cell, and that's really the crux of the technology. We know that it prevents graft versus host disease in a mouse model, and we believe that the reason that we don't see a high degree of GVH in our humans, obviously we can't test that scientifically as readily as we can in mice, is because of the tolerogenic nature of the cell. The product combines facilitating cells with hematopoietic stem cells through a proprietary manufacturing process, and the result is the ability to induce donor and host tolerance. The applications are broad. Really, any disease that can be treated by a bone marrow transplant, a safe hematopoietic stem cell transplant, can be treated with FCR001. And the major indications that we have elected to focus on initially are the solid organ transplant recipients, the life-threatening autoimmune disorders, including the rheumatologic disorders, which are scleroderma, lupus, and rheumatoid arthritis, and also Crohn's colitis. It's very interesting in that it's very much accepted now that bone marrow transplantation can result in a halt of disease progression in autoimmune disorders. We have compelling data from our Phase II Living Donor Kidney study, where all of our subjects who had an underlying autoimmune disease that resulted in loss of kidney function have not recurred in the patients who are durably chimeric. And in the long-term pipeline, we also potentially intend to focus on leukemia, the hemoglobinopathies, and the rare childhood disorders. The most mature indication we have, I'm a transplant surgeon by training, and so we focused our initial technology on solid organ transplant recipients. And for those of you who don't know somebody who's had an organ transplants are life-saving. For kidney failure, it's much better than dialysis, but patients face real challenges. They have a fixed loss of kidney function due to the toxicity of the drugs after transplant. There's a higher rate of malignancy. Diabetes and hypertension are major complications that result in about 50% of patients dying by 10 years. So we're saving lives with kidney transplant, for example, but it isn't perfect. And the way to make it even better would be to eliminate the need for the toxic immunosuppressive drugs. The cost to the payers is significant. Each rejection episode costs approximately $50,000. And clearly, if there were a way to prevent the use of the immunosuppressive drugs, it would be a major advantage. We have now transplanted under a Phase II study 37 subjects. The majority were performed at Northwestern. We did one patient also at Duke University to show that it wasn't limited to a single site. The safety profile of the conditioning and the FCRx infusion has been deemed to be acceptable. 26 of our 37 subjects are completely off immunosuppression for up to 95 months now. None have had to go back on immunosuppression. None have developed rejection episodes, and none have developed anti-donor antibody. And those are the main parameters we as clinicians follow in renal transplant recipients. After we did adjustments in how we manage the patients, and I can show you the slide showing that next, we actually, our success rate in getting patients completely off immunosuppression is 82%. And of those patients who are only transiently chimeric and not durably, there's still a beneficial outcome in that they need less immunosuppression than patients on standard of care. So even if it's a quote-unquote failure, it's really not a complete failure if you can get a patient down to one drug instead of two. The chimerism is not dependent on HLA matching. The majority of our patients have actually been highly mismatched. And the durably chimeric subjects who are off immunosuppression have had no evidence for rejection on protocol biopsy. Whereas you'll see in a couple slides later, there's a significant percentage of patients on standard of care immunosuppressive treatment with two drugs who show very mild changes of rejection on protocol biopsy. We've had no autoimmune disease recurrence in the durably chimeric subjects, and we've shown that the patients are immunocompetent to respond to vaccination. And this just shows, this is on a patient-by-patient basis, and the green shading shows when the patient has come off immunosuppression. At point number 1, we excluded patients who were sensitized, and we test a panel reactive antibody, and a PRA greater than 20 indicates sensitization. So after we had two patients who did not establish any chimerism who had a PRA greater than 20, we changed our exclusion criteria to exclude patients who had a PRA greater than 20. At time point number 2, we changed from managing the patients as we would for a typical kidney transplant patient to a hybrid of how you would manage a bone marrow transplant patient and a kidney patient, and that was when our success rate went up to 82%. And this just shows the degree of mismatching and the number of patients who were enrolled with each degree of mismatch. The off immunosuppression group is the group who are durably chimeric. The single immunosuppression group is the group who are transiently chimeric. And you can see that the majority of those patients were highly mismatched, and that did not impact the rate of success that we had for establishing chimerism. We compared a group of standard of care patients transplanted at Northwestern at the same time period to our tolerant patients. And the good news from the conditioning is that our patient and graft survival were similar. And for those of you who aren't familiar with kidney transplantation, the early results in kidney transplants up to about 5 years are really, really good. And so the fact that our graft survival and patient survival are similar means that we're not doing significant additional harm with the conditioning and the FCRx transplantation. What really was striking to us was when we looked at the kidney function, the glomerular filtration rate, how fast the kidney filters and how efficiently, there was a highly significant difference in superiority in our patients compared to standard of care. And you can see in the transiently chimeric patients, it was sort of in the middle. So, but a difference of 2 is considered significant, and ours was a difference of 25 for the GFR. We also looked at the major causes of morbidity and mortality, which is hypertension and hyperlipidemia caused by the immunosuppressive drugs. And essentially, in our tolerant subjects, there was about the same rate as in normal healthy patients, 18% hypertension and 9% hyperlipidemia, whereas in our patients who, the standard of care patients, 83% had hypertension because of the drugs, and 43% had hyperlipidemia. And when we looked, actually, I don't have the data here, but when we looked as early as 2 years out after the transplant, those differences were already emerging. This is our first patient who was transplanted. He's now almost 10 years off immunosuppression. He has 4 children and says that he's never felt this good in his entire life. His family has polycystic kidney disease, and that's a disease where you start feeling ill when you're a teenager because you get cysts in your kidneys and your lungs and your liver, and they get infected and you start to feel ill. And he did not tolerate dialysis at all, and actually his neighbor was his donor. And he essentially, once he was completely off immunosuppression, he says he can't remember ever feeling this good. And one of the other patients that we're really excited about is one of our tolerant patients who is chimeric, just actually 8 months ago now, delivered a healthy full-term baby. And for those of you who know transplant recipients, normally patients who have babies who are on immunosuppression are considered high risk, and they tend to have low birth weight babies and premature babies. And so this is an N of 1, but we were really pleased to see this baby come through a successful pregnancy and delivery. And it's the patients that we're all about. And so we look forward to embarking on the phase 3 pivotal trial and also expanding to the autoimmune diseases. And in summary, in terms of takeaway, in the phase 2, we've transplanted 37 subjects. Our graft and patient survival are similar at 2, 3, and 5 years. 70% of subjects that we've transplanted have been removed from all immunosuppressive therapy at 12 months following transplant, and none have had to go back on immunosuppression. And the initial analyses of our cohort compared to the standard of care showed a superiority that began to be apparent as early as 2 years after the transplant.